Okay, 4.5 is our study of our ecosystem. So we know our ecosystem is our interaction of our organisms and their environment. So first steps that you may have done, so you might have done a woodland, a grassland, seashore. I'm mainly going to be talking about a woodland. So first thing that you will do is you may go out and you'll map your ecosystem, and within that you'll identify 3 habitats within that ecosystem. You'll identify flora and fauna, so you're going to have to identify 5 plants, 5 animals. You'll conduct one quantitative survey on flora and one quantitative survey on our fauna. You'll present your information, you'll note and measure 3 abiotic factors, and you're going to identify any sources of error that may have occurred there. So, mapping of our habitat, so your woodland or grassland. You may draw a sketch of the area that you're studying. You include features such as trees, bushes, so it might be there, so you can see here that it's a fence drawn in here. So like if you had a tree, this could be a large tree, this could be a small tree. Okay, so when studying an ecosystem, we're going to select habitats within it. So the more habitats you study, the better. So if possible, have at least 3. So I would look at, with my students, ground view, litter view, and then the soil. Of that, you have to identify 5 flora and 5 fauna, and I would keep it really simple. So flora are your plants. Okay, so they may ask about flora, and fauna is anything related to the animals. So here are some examples of plants and animals that would come across in a grassland or a woodland. So dandelion grass, we have our clover, nettles, daisy, dockleaf, buttercups. And then some of our animals, we have ladybirds, caterpillars, butterflies, spiders, slugs, beetles, snails. Now, of that, we need to know any adaptations of those plants or animals that will help them to survive. So remember, an adaptation can be structural or behavioural. So improve an organism's chance of survival and reproduction. So here's just 2 examples I have of 2 animals and 2 plants. Ladybirds. So ladybirds, they're brightly coloured, okay, and then the benefit of this is avoids predation, as bright colours, not birth colours, indicate acidic toxins in nature. And sparrowhawks, we mentioned earlier, they have sharp talons, will help them to catch their prey. With plants, dandelions. So dandelions, they have seeds have parachutes, okay, so that means they're easily dispersed when they dry out, so they're not brightly yellow. And then nettles have stinging cells that produce an acid to deter consumers as well. Now, if we're asked to identify an animal or plant, we don't know how to, what you use is you can use a key. Recently, when this is asked, they also accepted an app. There's apps out there. So what is a key? So a key is a means of naming organisms by answering a series of questions with alternate answers. So they have asked before to draw a key to identify for a flora or fauna. So here's a very, very simple example. So we have our animal here. So are wings present or are wings absent? So if wings are present, we're saying it's a housefly. If wings are absent, so then we have to look at the next thing. If you're saying no, there's no wings present. So how many legs does it have? Does it have 8 legs? So more than 8 legs. So if it does, we're going to go on to the next step. If it has no more than 8 legs, then it's a spider. So we're saying if it does have more than 8 legs, 1 pair of legs per segment, so it could be a centipede, and 2 pairs of legs. It could be a millipede. So we have to be able to collect animals and plants, and we have to know 5 methods of collection. Okay, so here we have our pewter. Our pewter is used to collect insects and spiders. So you're going to suck the mess through one tube, and at the other tube, you're going to put that over the organism. A beating tray is another example. So with a beating tray, can be a white sheet really actually as well. So you go over to a tree or a bush, you get a stick and you hit it 3 times. The first time you've shocked the insects, what's there. The second time they're trying to cling on, and the third time they end up falling off. And we use a white sheet, so a white sheet that we're able to identify the organisms. If we're not able to identify them, we use a key. So insects and caterpillars, maybe some spiders. A pitfall trap is another example there. So a pitfall trap, you might get a glass jar and you dig a hole within the soil. Okay, place your glass jar so that your glass jar is level with the soil. You would then put maybe some rocks or stones, something around it with a cover on top. And we put a cover on top because if it rains and there's any insects in there, they're just obviously going to drown, and we don't want to do that. So insects, we may find beetles, well, may find them there, and you may find spiders. A sweep net, so you may use a sweep net, so that would go through long grass. You'd run that through long grass, and then you can turn that sweep net, the contents of that, onto a white sheet. So again, we're able to identify what are the insects there. If you can't identify them, we use a key. Now, tulle green funnel as well is another. So with a tulle green funnel, you're going to collect a sample of soil, and you may bring that sample of soil back to the lab. And here we have our tulle green funnel set up. So, or you may have someone slightly different than this. So you put your soil contents inside here and you turn on the light source. And what will happen is if there's any insects that are buried in the soil type, they will bury themselves away from the light source. And here you'd have a gauze area, they'll fall down and in here into the alcohol underneath, and you're able to identify that, the organisms. But there's 5 that you need to know. If you could find a rock, go by direct search and go up and lift up a rock and you might find woodlice. So this leads us on to our quantitative and qualitative surveys. So we're estimating the number of flora and fauna within our ecosystem. So we need to know the definitions of qualitative studies, the presence or absence of organisms. And our quantitative records the number of organisms that are present. So it's important in carrying out qualitative and quantitative surveys. So it gives you an insight into the number of species in an ecosystem. It detects change in population numbers, and it can look at insight into the relationship between organisms and their populations. So to carry out a quantitative survey of plants, you would use a quadrat. So you might use a gridded quadrat here. It depends on what you have. Some of you might have a square frame with wires going across. So limitations can only be used for small plants, really. Fast-moving animals won't stay inside a quadrat, and trees, they're too big. So use the quadrat to measure the percentage frequency. So what's the first thing that you do? So the first thing that you do is you throw a pen or a pencil over your shoulder. Why are you doing that? One, it's random, and it's also reducing bias of where your pen has landed, and it's health and safety. You're not going to go and fling a quadrat. The next step then you have there is you're going to place your quadrat over when your pen landed. And then you're going to note if any plants were present or absent. So by this, you would have decided already, okay, I might want to look at grass, daisies, dandelion, clover, keeping it simple. So you're going to record your results. And then you're going to repeat this again 9 more times. So I throw my pen over my shoulder. Why? It's random, it's reducing bias, and it's safety. I place my quadrat down. I'm going to see if my daisy is present or absent. From that, you've done that 10 times. So then you're going to calculate out the percentage frequency. So of the percentage frequency, you've seen, for example, the daisies there 7 times out of 10, so 70% frequency there. You're going to present your results in a table, and then you may plot your results in a graph. So percentage frequency is the chance of finding a named species of a throw of a quadrat. A disadvantage can be dependent on organisms and quadrat size, or it can assume that organisms are evenly distributed. So here we have for percentage frequency. So daisy, I have there's actually 5 times. So 5 out of 10, 50% present. Dandelion is there 4 times. The X means that on this, for example here, on the 3rd throw, there was no dandelion or daisy present within that quadrat. So, and how do I present my results? Here's my results presented in a bar chart. Okay, you could also do this using a line transect. So a line transect is a rope, and on the rope there's intervals, which could be every metre. So you're going to record what touches the line transect. And ideally you'd have it started off that you'd cover like where it was bright, some in the shaded and some in dark. So it's not random as you choose where to place it, that's the only thing. Now percentage cover. With the percentage cover, it's an estimate of the amount of ground a quadrat is covered by each species. So this is where we're going to use our gridded quadrat. Okay, so you might have used your gridded quadrat as well, but we're just going to go into our gridded quadrat a bit more detail. So again, same thing. I'm going to throw my pen over my shoulder. Why? It's random, it reduces bias, and it's safety. I'm going to place my quadrat down over where my pen is, and I've already decided what the plant is that I'm looking for. So you're going to place the quadrat over the pen. So I have here daisy and dandelion again. So you're going to push the pencil down on the gridded points. So it could be the points here on the right hand side that you're looking. Is the daisy present? Yes or no? Okay, that's what you're looking for. So you're not looking to see if it's in the box area, you're looking to see if it's in a point. So you're going to record your results. Again, repeat this 10 more times. So you're going to calculate out the percentage. You're going to put your results in a table, and you're going to graph your results. So here we have Daisy again. So the total number of hits in that corner was 40. There's 250 points because there's 25 of those little squares, and we've done this 10 times. So we can represent our results here in a bar chart. You then may be asked to find out a quantitative survey of animals, and I use the examples of a snail. So most animals are fast moving, so they need to be quantified using the capture-recapture method. So an example that I use with my students is the snail. So we would go out, and I would do an activity of this that we're not actually looking for snails, but something else. So we would go out on our first day. And on the first day, we go to our habitat. So on our first visit, we're going to search for snails. And we're going to count how many snails that we have. So we search for the snails, and we're going to mark on each of their shells with example for a Tipex. Something that is non-permanent. Okay, so we don't want to be drawing attention to them. So it's important that the mark does not harm the snail or make the snail more visible. So we're going to record the number found and marked, and we're going to place those snails back. So that's day one, that's visit one. The second visit is going to be approximately 2 or 3 days later. It's not going to be a week later or longer, because if it is, the snail more than likely may have been consumed by somebody else or moved further away. So on the second visit, we're going to return to the same habitat, and we're going to search for the snails again. So you're going to note how many of the snails are found, and of those found, how many are recaptures. So how many may have the Tipex on their back. And then you place the snails back exactly where you found them. Okay, so then we're going to calculate out the population size. So to do that, I have there capture 1 by capture 2. So capture 1 is the number caught and marked on the first day. So I have example here. On the first day, for example, I caught 40. And of the 40 snails, I put Tipex on their back, each of their back. And then on my second day of capture. So it's the number caught on the second visit. So I'm saying on the second visit, I caught 40 snails again. But of those, 25 had Tipex mark on their back. So there we have 40 by 40 over your 25. Calculate that out, and that will give you the number of animals in your habitat. So that example would be for snails. So then we're on to measuring abiotic factors. So we need to measure 3 abiotic factors. So I've got 4 examples here. So some of you have done woodland or grassland, you may have done these as well. There are other ones that are just not within the notes. So ground temperature, if I was doing ground temperature, I have a thermometer. So here's my ground thermometer. I'm going to place it in the soil. So I'm going to place it in the soil, record the temperature, and I'm going to go to 2 other areas. So I'm going to get an average. And what we should find with the temperature, it affects the metabolic rate of what organisms grow there. So higher temperature in summer, so rapid growth of plants and high levels of animal activity. You may then do air temperature. So air temperature, I have a thermometer here and I have it hanging off a tree. And I'd go record it in 3 different areas. And you may find that high temperature, low temperature again. So for example, low temperature in winter, many animals may hibernate or death for many. So both is air temperature, so both we can use the thermometer here as well. So I'm just going to say T for thermometer can be used here as well. But what's the influence of the factor? Okay, so then another option that we can look at, I like doing, is soil pH. So I take a sample of the soil, you can put it in a pH monitor and take a sample of this and record that. And then go to 3 other areas because we want our 2 other areas. So we have 3 areas, so now we get an average. So we find then, so favours some plants and therefore some animals, depending on what the acidity of the soil is. Then you may do a light intensity. So you may use a light intensity meter. You'll hold it up to take the top off, hold up towards the light, record the reading, and go to 2 other areas. So plants are adapted then to different light intensities. So how do we present the information that we've done on our field study? So we may use charts, tables, graphs, diagrams, or histograms to represent. We'll have to analyse the relationships between the organisms, including our food chains, food webs, and food pyramids. So we've already covered a food chain, we've already covered food webs, and we've already covered pyramids of numbers. So they can be linked in with this. Now, they may ask you about sources of error, and the two that I would teach my students is miscalculation, very easy. So you calculated up something wrong. You don't need to let them know, say you calculated up something wrong. If you ask for a source of error, miscalculation. A second one is misidentification. You didn't use the key or you identified the wrong organism. You don't need to say that. So just misidentification or miscalculation. I would know those two. Other possible sources of errors is changing conditions, so types of seasons, and there could be of sample size.